Okay, so what are we trying to do here? Once again, we are taking single genes, or in the case of prokaryotes, multiple genes, and we are going to turn the DNA code into a messenger RNA code. And again, it's not going to be the whole piece of DNA, the whole chromosome. It's just going to be a gene if it's a eukaryote, or potentially several genes in a prokaryote. And the reason why this happens is because the DNA is protected inside of the nucleus. Well, where the ribosomes are located, and that's what's going to turn the nucleic acid message, in this case it's going to be messenger RNA, into a functioning protein that's occurring in the cytoplasm. And the cytoplasm has got all sorts of enzymes and things of that nature that could degrade or break down the DNA. So why send out that $1 million blueprint, which is huge to start with, so it couldn't leave the nucleus anyways? Instead, send out a cheap, recyclable, disposable copy of just one or several genes in the form of messenger RNA. Okay, so the process of transcription, RNA polymerase is going to be copying the DNA template strand. So in order for that to happen, okay, a factory, if you will, consisting of enzymes and proteins has to assemble in the right location on the DNA. What is the right location? That's the gene that has to be transcribed. What determines what gene's being transcribed? That's going to be different types of transcription factors, which are proteins that are going to assemble at the site of the beginning of a particular gene. We're not going to get into the details how that all happens right now, but you will have to know that in front of a gene is an area we call the promoter. And the promoter is the site where this transcription factory is going to assemble. Okay, so as you look through this diagram on the picture here, you can see the promoter site with this little TATA box. That's just a series of adenines and thymines. Okay, and then a transcription factor will anchor onto that TATA box, and then other transcription factors and the RNA polymerase will then assemble onto that promoter region. And once everything is in place, then the process of transcription can begin. And again, remember guys, this is all occurring in the nucleus. This is a pretty complex process, and we're just doing a pretty cartoonish overview of this process. So that was initiation. The second stage is called elongation, and that's going to be the addition of nucleotides to the growing messenger RNA strand. Now remember, we can only add nucleotides to the 3' end because that's where we can form a bond. Remember, we're going to form bonds, covalent bonds, okay, by dehydration synthesis, and this is going to be controlled by RNA polymerase, which is an enzyme, and we can only add on to that 3' end. So let's look carefully at this diagram. We see the newly made RNA coming out in pink there. If you notice, it's the 5' end that is leaving the RNA polymerase, where the 3' end is where we are adding new nucleotides to. So which strand is being copied? Well, if you look at the DNA carefully, you can see that the DNA strand that is being copied goes 3' to 5'. You can see the 3' on the top on the left-hand side in the blue DNA molecule, and that's the one that's being copied. Okay, and again, it's being copied to base pair rules. Notice once again that RNA does not have thymine. It has uracil in place of thymine, and A is pairing with U. Okay, C and G always pair together. And so this RNA polymerase will continue moving down the DNA adding nucleotides to that 3' end until it reaches the termination site. And the termination site is simply going to be a stop codon. It's a series of 3 nucleotides that code for, hey, this is where you got to stop and you got to break off and see you later. Hasta la vista. Okay? And once that has been completed, then that pre-messenger RNA transcript, that's what it's called, the pre-messenger RNA transcript, has been created. Okay? So again, this is in a eukaryote. Eukaryotes, the pre-messenger RNA transcript has to undergo modification. And you can see there's a couple of modifications we can see here already. We can see that there's this 5' cap and a poly A tail. So what exactly are those? Well, the 5' cap is kind of a protective headgear, if you will. It's going to, it does 2 things. One, it's going to prevent the front part of that messenger RNA molecule from being degraded by enzymes. And number 2, it's going to play a role in directing that messenger RNA molecule to either a free-floating ribosome in the cytoplasm or to a ribosome that is attached to the endoplasmic reticulum. So it kind of guides it. It's kind of like a little guidance system. The poly A tail, which can be quite long in some cases, consists of a whole bunch of adenine units, and it plays somewhat of the same function as the 5' cap. It helps to prevent the premature degradation of the messenger RNA molecule. The longer the poly A tail is, the longer the messenger RNA molecule will remain in the cytoplasm being translated into proteins. The shorter it is, then it won't last very long in the cytoplasm, and it will only make a limited number of gene products. But if it's really long, it will make a whole bunch of the gene products. The other thing that has to happen, and this only occurs, remember, in eukaryotes, is that we have to get rid of the DNA that does not code for protein products. Now look at the top of this diagram. We can see that the pre-messenger RNA molecule consists of exons and introns. Exons are coding DNA. They are going to be expressed into an amino acid sequence. Introns are non-coding DNA, and they have to be cut out. We have to remove the introns. I'll get into the functions of those introns in class, or when I see you guys in AP biology, we'll talk about the function of those introns. But for now, I'm not going to dive into those details. But just suffice it to say that we have to get rid of that non-coding DNA called introns. And as you can see there, our messenger RNA molecule has the 5' cap, our poly A tail, and no introns. They are gone, and all the exons have been spliced together. So how do we do that? How does your cells take care of that out? Well, they use a special combination of proteins that are grouped together along with some RNA molecules that we call SNPs. That's the SN RNA. There's another type of RNA molecule. Those things all will come together at the site of an intron, and they'll self-assemble into something called a spliceosome. Soma means body, and splice means to cut apart. Now, you guys are too young to ever have to splice a cassette tape, but back in the day when we wanted to make a mixtape, oftentimes we had to take the tape and we had to cut out the bad pieces of tape if we made a mistake or if it had too much static, and we'd have to splice just the good music together. And so that's where splicing comes from. Or sometimes if you do a lot of rope work, you have to splice 2 ropes together and stuff. So anyways, what a spliceosome does is it excises or cuts out the introns and splices together the exons. Okay, so that's part of that post transcription modifications that has to happen, and this is all occurring in the nucleus before it leaves the nucleus. Okay, so here it is, kind of a big picture of it. We got the transcription occurring in the nucleus. We make the pre-messenger RNA. The pre-messenger RNA undergoes modification to make the mature messenger RNA, and that's going to go out into the cytoplasm where it will attach to a ribosome in order to make a functional protein. Now, if you look at your notes also, you can see that, and now this is on the back page of your notes, is that these exons can be spliced together into different combinations. We as humans have only about 22,000 genes, but we can make hundreds of thousands of different proteins. Well, remember Beadle and Tatum came up with the idea of one gene equals one protein, but that's not quite true. We now know that one gene can be modified into a variety of different types of proteins depending upon how we splice the exons together. And there are other types of genes that don't make proteins at all. They make RNA molecules. Okay, different types of RNA, like the SNPs that we just talked about, or another one called RNAi, which plays a role in shutting down DNA of invading organisms and things of that nature. Okay, so that's kind of part 2 in a nutshell. Hope you enjoyed it. Take care, and we'll see you in part 3.